Okay, okay pa?
Okay, okay po, Doc.
Okay pa po, Doc.
All right. So this afternoon, we'll just go all over the exam. Now,
Yes po, Doc.
I want you to take note, especially the questions on the exam. All right. The test, the first is matching, di ba? No? You have matching. Okay, the first one was water and acids. Remember? And you are to select the 4 options: pure water, hydrochloric acid,
And hydrochloric
both
acid.
water and hydrochloric acid, and neither of the two.
This
Okay? All right, list number 1. Which of the two exhibit tendency to dissociate?
Both.
both.
Both. Both. Remember that? No, both. No, of course, acid dissociates. But water also dissociates, but not. There is a small amount of dissociation. Only a very small amount. Okay. Number 2. The dissociation constant is 1 times 10 to the minus 14.
Water.
Water. Okay. Or water. Number 3, a pH below 7.
Hydrochloric acid.
pH below 7.
HCl.
Hydrochloric acid. Okay. Hydrochloric acid. All right. Number 4. Its conjugate base is bicarbonate.
None of the above.
None. Okay, none. Number 5. Can dissociate completely.
Hydrochloric acid.
Okay, because hydrochloric acid is considered to be a strong acid. Now, strong acids or strong alkalis, they dissociate completely. Okay? So, who got perfect in that water and acids? Huh? Nobody? Huh? Sharo naman sad. Okay, let's go to the next one. Major causes of diseases. That's the first chapter, di ba? All right. To have the diseases here or disorder, scurvy, sunburn, sickle cell anemia, morphine intoxication, and hoof and mouth disease. All right, number 1. Which one is a physical agent?
Sunburn.
Sunburn. Okay. Number 2. Nutritional imbalance.
Scurvy.
Scurvy, vitamin C deficiency. No? Vitamin C deficiency. Number 3. Genetic disorder. Sickle cell anemia. Okay, sickle cell anemia. What is the anomaly there? Substitution of an amino acid. No?
Point mutation
Of your beta subunit, the number 6 amino acid, which is glutamic acid, and replaced by
Valine.
valine. All right. Always remember that. no? That will come out in your board exam. Not in medicine, not in biochemistry, but perhaps in medicine. All right, number 4. Chemical agents.
Morphine.
Morphine overdosage. Morphine overdosage. Okay. Biologic agents.
Virus.
Hoof and mouth disease caused by a virus. All right. So, everybody got perfect there? Those 5 items? Wala.
Yes,
Yes, Dad.
yes, Doc.
Taas ang kamay. All right. Next one. Proteins. Your choice, collagen, myoglobin, hemoglobin, thalassemia, chaperone protein. All right, number 1. Function in carbon dioxide and protein transport.
Hemol-
Which one?
Hemoglobin.
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 Yes. Klaro na ko na, transport. There's only one transport there, that is hemoglobin. Diba? Number 2. Brought about by reduced production of alpha or beta subunit of hemoglobin.
Thalassemia.
Thalassemias. All right. Number 3. Requires ascorbic acid for its proper synthesis.
Collagen.
Collagen. Okay. Number 4. Assist in the proper folding of over half of mammalian proteins. Chaperone proteins. Hindi na uso ngayon ang chaperone, no? Pag mag-date kayo, wala nang chaperone.
Bawal chaperon.
Yung sa amin, mayroong chaperone. During our time, mayroong chaperone. All right, number 5. Oxygen binding curve is hyperbolic.
Bin.
Myoglobin, hyperbolic. Hemoglobin, what is the curve?
Sigmoidal.
Sigmoidal. All right. So who got perfect 15? Wala pa? All right. Next one. Oh, no, by the way, this collagen, myoglobin, hemoglobin, thalassemias, and chaperone protein, this particular matching set, there is something wrong here. No? Because thalassemia is not a protein. It's a disease. Di ba? You get it? No?
Yes.
You must be getting old. Alright. Next one. Amino acids. You have cysteine, serine, glutamine, methionine. Okay, so number 1 contains a phenyl group.
Phenylalanine.
Phenylalanine. O nanaguna, phenyl group, phenylalanine. Giveaway na, no? Number 2 has an OH group, hydroxyl group. Serine. Serine. All right. Number 3 has ethyl group,
Cysteine.
cysteine. Now, this spelling of cysteine there is not correct. If you remember, the spelling of cysteine is not correct. It was wrong.
E.
All right. Number 4 contains an amide group.
Glutamine.
Glutamine. All right. Contains a thiomethyl group.
Methionine.
Methionine. All right. Aside from serine, what amino acid contains a hydroxyl group?
Threonine.
Threonine. Threonine. Tyrosine. Okay? Threonine, tyrosine, and serine. They have hydroxyl group. All right. Aside from glutamine, what other amino acid contains an amide group?
Asparagine.
Asparagine. All right. Okay. Let's go to the next one. We have now the structure of proteins. You have primary, secondary, tertiary, quaternary. Okay? All right. Number 1 requires the presence of more than one polypeptide chain.
Quaternary.
Quaternary. Okay. At least 2 or more subunit or polypeptide chain. This is the quaternary refers to the non-covalent interaction of subunits. Non-covalent. Remember that. So what is holding them together are the non-covalent interactions. So what are those non-covalent bonds?
Hydrogen bond.
Which bonds? Ionic.
Disulfide,
What else? Hydrophobic. And a very weak force, van der Waals.
Underwells.
All right. Number 2 is formed by linking one amino acid to another by peptide bonds.
Primary structure.
Okay, primary. Primary structure is formed by linking amino acids by peptide bond. Number 3 is produced by the interaction of side chains of the amino acid.
Tertiary.
Tertiary. Okay. Tertiary structure is made by the interaction of the side chain of your amino acids. That is your tertiary. Number 4 results from the formation of hydrogen bonds parallel to the axis of the protein.
Secondary.
Secondary. Which of the two secondary structures? There are two there in secondary.
Alpha.
Alpha helix. All right. Number 5 is ultimately dependent upon the sequence of amino acids in the protein.
Primary. All.
What's other opinion?
All of the above.
All of the above.
All of the above.
All of the above.
Yay.
Mm,
Although the primary structure refers to the sequence of amino acids in the protein. No? The coiling, the folding, and subunit interaction will also depend on the location of the amino acid in the polypeptide chain or the sequence of the amino acids. So it is also dependent. Diba? Even if you are in the quaternary, you are still dependent on the primary. Correct? All right. Next one. Alpha helix, beta pleated sheath, random coil, all of ABC, none of ABC. All right. The first question is, structure is broken at proline residues.
alpha, helix,
Alpha.
alpha.
Alpha helix. Alpha helix. All right. Alpha helix, proline is an amino acid. Proline does not form hydrogen bonds, which is necessary for maintaining your alpha helix. It cannot form an hydrogen bond, your proline. If you look at your proline, you'll have a hard time for your hydrogen bonds to form. Okay? Number 2 is produced by procedures that denatures protein.
Random coil.
All right, random coil.
Random coil.
Random coil. Denaturation. Now, by the way, how do you denature the protein?
Heating, heat,
Heating is one.
heating,
Yes. Heating is one.
pH,
Seeds.
pH. What else?
Ultraviolet light.
Salt solution. No? You can denaturize your protein. All right. Denaturation disrupts the non-covalent bonds that maintains the three-dimensional folding of the polypeptide chain. Disorganized conformation of the polypeptide chain is called random coil. The protein usually loses its biological activity when it is denaturized. Well, it should also be noted that undenatured protein, meaning it has been denaturized, may be in a random coil, aside from the alpha helix and the beta pleated sheath. Okay? All right. Number 3 is present only in proteins that have a quaternary structure.
None.
none of the
None.
above.
None po.
None. Okay. All right, number 4 is maintained by hydrogen bonds parallel to the axis of the protein.
Alpha, alpha
Alpha
helix,
helix. That was a question before. Diba? In the primary structure. No? The primary structure, secondary structure, tertiary structure, quaternary structure. Okay. That's number 4 question. Results in the formation of hydrogen bonds parallel to the axis of the protein. And then you have it here again, is maintained by hydrogen bonds parallel to the axis of the protein. Huh? All right. So you should, if you answered your letter B in the first set, secondary structure, you should be able also to answer that in the succeeding set. Because this question refers to the secondary structure. All right, number 5 is formed by hydrogen bonding between the peptide bonds on adjacent chain.
Beta pleated sheets.
Beta pleated sheath. All right. The hydrogen bonds there are perpendicular to the axis of the chain. And they can occur between parallel or anti-parallel polypeptide chain. Huh? Okay. Next one. Vmax and Km. So we have both A and B, neither A nor B. So this is enzyme kinetics, which is independent of enzyme concentration.
Both A and B.
No, it's Km. It's Km. You should remember that an enzyme can be identified by its Km. What is Km? What is Km?
With substrate to reach 1/2 Vmax.
Km is the amount of
Substrate.
Substrate.
substrate concentration
Needed to reach 1/2 Vmax.
to be able to reach 1/2 Vmax. Okay. Now, each enzyme has its own Km. Even if you increase the substrate, the Km is still the same. Because enzymes are saturable. Once it is saturated, it cannot act on the other substrate. It only acts on the other substrate when the substrate that it is attached to it has been broken down into products. So the catalytic site becomes empty. And when it becomes empty, it can now be able to bind another substrate. Can you get that? All right. Number 2 is independent of substrate concentration.
Vmax.
Vmax.
Is independent of substrate concentration.
Both A and B.
Both of them. Both of them. Number 3 is independent of temperature and pH of the reaction.
Neither.
Neither. None of them. Huh? Neither. Number 4 is related to the slope of the Lion-Weber-Burke double reciprocal plot.
Both.
Both of
They,
them. Both of them. A and B. Number 5 is directly equal to the reciprocal of the y-intercept of a Lion-Weber-Burke double reciprocal plot.
Vmax.
Max. All right. So there you are. That's your enzyme kinetics. Next one. Inhibitors. You have two here. Competitive and non-competitive. Number 1. Occurs when succinate dehydrogenase is inhibited by malonate.
Competitive.
Competitive po.
Competitive. All right. Succinic acid is inhibited, succinate dehydrogenase is inhibited by malonic acid because malonic acid resembles succinic acid. So it can bind to the catalytic site. Okay? Number 2, binds enzyme at sites away from the active site.
Non-competitive.
Non-competitive.
Non-com.
So if it binds to the other site, other than the catalytic site, so how does inhibition comes around? All right, the moment the allosteric, we call it allosteric. Allosteric meaning others, no? Other sites. The allosteric inhibitor, once it binds to the allosteric site, it changes the shape of the catalytic site so that the substrate cannot bind to the catalytic site since it changes its shape. So there is no binding. Okay. Number 3, results in an increase in the apparent Km of the reaction.
Competitive.
Competitive. Competitive. Because to remedy the inhibition of a competitive inhibitor is to increase your substrate concentration. So by increasing the substrate concentration, you have more or less a good chance of having the enzymes to be able to bind to the substrate than to the competitive inhibitor. Because you are increasing the number of molecules of your substrate. So chances are your enzyme can bind to the substrate. In that way, you increase the Km of the reaction. Okay. Number 4, has the same Vmax as the uninhibited reaction.
Competitive.
Competitive. No? Vmax is still the same.
Competitive.
Yes. Has the same Vmax. Only the KM is affected by competitive inhibition, but not the Vmax.
Com-
Yes. Competitive. Competitive has the same Vmax. What is altered is the Km. That was our number 3 question. Results in the increase in the apparent Km of the reaction. When you say increase of Km, that means more substrates are needed. Di ba? All right, number 5. Usually involves inhibitors that are structurally dissimilar to the substrate.
non.
Structurally dissimilar.
Non-competitive.
Non-competitive. All right. Let's go to the next one. Enzymes as diagnostic molecule. You have there alanine aminotransferase, amylase, ceruloplasmin, lactate dehydrogenase isozyme 5, and alkaline phosphatase. Number 1, Wilson's disease or
Ceruloplasmin.
Plasmin.
ceruloplasmin.
Zero.
Okay, ceruloplasmin. Number 2, viral hepatitis.
Healthy.
Alanine aminotransferase.
Alanine aminotransferase or ALT. Liver disease.
T. Dehydrogenase.
LDH.
LDH5. Lactate dehydrogenase isozyme 5. Bone disorders.
ALP.
ALT.
Alkaline
AL.
phosphatase.
Phosphate.
Alkaline phosphatase. Acute pancreatitis.
Amylase.
Amylase.
Yes.
Amylase.
All right. So next one. Vitamins as precursors of coenzymes. We have thiamine, vitamin B1, no? Niacin, riboflavin, vitamin B2, pantothenic acid, and vitamin B6. If you notice, they belong to the vitamin B complex family. All right, number 1, FAD,
Riboflavin.
Flavin.
riboflavin. Okay. That is flavin adenine dinucleotide. Number 2, coenzyme A or CoAs, pantothenic
And
acid. There's a shampoo that contains pantothenic acid.
Pantin.
Very good, Pantene. So you have been using Pantene, ha? All right. Number 3, NADP,
Nias.
niacin.
niacin.
Niacin, nicotinamide adenine dinucleotide. The nicotinic acid from niacin, no, is amidated and becomes niacinamide, and then interacts with ATP to become adenine dinucleotide. Okay, that's NADP. Nicotinamide adenine diphosphate. Number 4, pyridoxal phosphate,
Vitamin B6.
B6.
vitamin B6, pyridoxine. That's why that's PP, pyridoxine phosphate. Number 5, TPP,
Thiamine.
Thiamine.
thiamine pyrophosphate. No? This is quite easy because there's thiamine there, say TPP. So the first letter there. And also pyridoxine for vitamin B6, pyridoxine becomes pyridoxal phosphate. Niacin, nicotinamide. No? Madali ra.
Ikot.
Letter N. Okay, so let's go to multiple choice. In biologic membranes, still remember, and you just visualize biologic membranes. Proteins and lipids interact with each other by which of the following? Covalent bonds, hydrogen bonds, hydrophobic interactions, none of them.
Hydrophobic interactions.
Hydrophobic interactions. Remember that you have a protein and then you have lipids. Now you know that the protein is made up of amino acids. And the amino acids, if you still remember, you have polar and non-polar. Di ba? Polar and non-polar amino acids. So the protein that interacts with the lipids are those proteins which have an area in the protein molecule which is made up of non-polar amino acids. Because lipid is non-polar. So the interaction would be non-polar or hydrophobic interactions. Number 2. So the answer is letter C. Now, when I went over the exams, I noticed that most of the correct answers landed in letter C. I was very careful not to have the answer on letter A. And I was successful because there were only 4 numbers which has letter A as the correct answer. And then I went against the ruling in making questions that says that in making multiple choice question by not putting your correct answer at letter C. Now I notice I put more answers on letter C. Imagine, letter C has 15 correct answers. Letter C.
C for Christ, Doc.
Letter B is 12. Letter D is 9. And letter A is only 4. No? I must be sleepy when I made this question. All right, let's continue. Membrane proteins that span the whole width of the membrane are known as plasma proteins, letter A. Letter B, integral proteins. Letter C, nucleoproteins. Letter D, peripheral proteins.
Integral
Integral.
protein.
Integral
Integral.
protein.
So that portion of the integral protein, because it penetrates the whole membrane, no? That area of the protein that penetrates the membrane are made up of non-polar amino acids so that it can have hydrophobic interactions with the lipids. Otherwise, it would be rejected by the membrane if it is polar or water soluble. You know that the lipids are not water soluble. Okay? Number 3, selective permeability of cell membrane is achieved in part by active transport. And active transport differs from passive transport in that A, requires energy but no transport carrier. Letter B, is necessarily associated with pinocytosis. Letter C, it depends primarily on diffusion and osmosis. Letter D, it requires energy, usually in the form of a phosphate anhydride bonds.
Letter D.
Letter D.
Letter D is the answer. Huh? Letter D. All right. Number 4. Some antibiotics act as ionophores, which means that they inhibit both translation and transcription. Letter B, have detergent-like effects on cell membranes. Letter C, interfere directly with bacterial cell wall synthesis. Letter D, increase cell membrane permeability to specific ions.
D.
Letter D.
Letter D, no? Increase cell membrane permeability to specific ions. Okay? Number 5. The principal cation of the intracellular fluid is sodium, potassium, calcium, magnesium.
Sodium.
Potassium.
Potassium.
Intracellular. Extracellular,
Sodium.
Sodium.
sodium. All right. Number 6. The dominant phospholipid in the membrane is phosphoglycerides, phosphoglyceride or glycerophosphate. No? Phosphoglyceride or glycerophosphate. Number 7. Lipid bilayers are formed by chaperone assembly, guided assembly, self-assembly, none of them.
Self-assembly.
Self-assembly.
They are chaperone assembly, guided assembly, self-assembly, none of them.
None.
Self
Self-assembly.
ba, Doc?
Self-assembly.
Self-assembly.
Self-assembly. All right, number 8. Which of the following has a high permeability coefficient?
Water.
Water.
All right, that's water. We have here sodium, glucose, and water. Water has, among the three here, has the highest permeability coefficient. Number 9. The phospholipid marker of mitochondrial membrane is sphingolipid, cardiolipin, phosphatidyl inositol.
Cardiolipin.
Cardiolipin. Now, when you are going to do an assay of enzymes inside the mitochondrion, first you have to break the cells, di ba? Break the cell membrane, and then you have to break the membrane of the mitochondrion. Now, how would you know that you are already in the mitochondrion? You answered kanina cardiolipin, di ba?
Because of the marker po.
It's a marker. It's the one that will tell you that you're already breaking the mitochondrion. You know, we usually break the cells through what we call an ultracentrifuge. So we increase the rotation or the centrifugation, no, until we reach the point where we can detect the presence of cardiolipin. Because cardiolipin is plentiful in the cell membrane of the mitochondria. No, I'm sorry, not the cell membrane, the membrane of the mitochondria. Okay? The specialized structure of plasma membrane that prevents diffusion of macromolecules between cell is the cavuli, life rafts, tight junction, gap junction.
Tight
Tight
junction.
ba?
Tight junctions. Yes, tight junction. It's already there. Oh, sa question mo. Prevents the diffusion. So it prevents the entrance. So anything that is tight, you cannot penetrate, di ba? Boys. Can you relate to that, boys?
Kevin.
Of course, Doc.
Oh, na.
Opo, Doc.
So there is already there. Oh, the question says already prevents diffusion. So it must be tight. Come on, good eye. All right, the cross-membrane movement of large molecules is through which of the following? Endocytosis, passive diffusion, active transport, facilitated diffusion.
Indo.
Endocytosis.
Endocytosis. Okay, cross-membrane movement of large molecules is through which of the following? endocytosis. Number 12. Which of the following is not a property of ion channels? Letter A, most are highly selected for one ion. Letter B, their activities are not affected by drugs. Letter C, they are composed of transmembrane protein subunits.
Letter B ba?
Letter C,
Letter
Doc.
B.
Letter B ba, Doc?
Letter B. Ha? Letter C? Letter
Oy,
C is they are composed of transmembrane protein subunits. Letter B is the answer. Their activities are not affected by drugs. Because the question says is not. And there is already a hint there because letter B contains not also. Their activities are not affected by drugs. Calcium channels are affected by which drugs for hypertension?
Calcium channel blockers.
Calcium channel blockers. Naraman din na. Alright, number 13. These are proteins that form water channels in certain membranes. One is hydroprotein, B is ionophores, C is aquaporins.
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 Aquaporins. Now there are two here which refers to water. One is hydroprotein and the other is aquaporins. Diba? But we do not have hydroporins in the textbook. What we have was aquaporins. Alright. Number 14. Facilitated diffusion, letter A, can operate bidirectionally. Letter B, always against concentration gradient. Letter C, always utilizing energy.
always against concentration gradient.
Always against concentration gradient. Number 15. Cytochrome P450 are referred to as hydrolase
Monooxygenase.
Oxygenase.
Monooxygenase.
monooxygenases. Okay, very good.
Hi, Doc, excuse me.
Monooxygenases.
Yung
Yes.
14, di ba, diffusion, facilitated diffusion.
Facilitated.
Bakit po, Doc, against a concentration gradient?
It's always against concentration. That's why you have to have a facilitator. May simple diffusion, no? Pag tumaas na ang concentration on the other side, humihinto or it stops. Now, when it is facilitated, there is something that facilitates it, the diffusion. So it can go against concentration gradient.
Habok do kay diri.
Doc, passive transport, it facilitated.
Facilitated diffusion.
Facilitated diffusion, Doc, is it not a type of passive transport?
Yeah, yes, it can be.
Okay, di ba, Doc, passive transport goes down the concentration gradient?
Oh, that goes down, but it is not against.
From high
It is
concentration.
with. It is with higher concentration going to uh, low concentration. All right, number 15. Cytochrome P450 are referred to, okay, monooxygenases. You answered that already. Number 16. The main reaction in phase 1, metabolism of xenobiotics in the
Hydroxylation.
liver is hydroxylation. Oh, hydroxylation. Hydroxylation, just to make the molecule or the xenobiotic more soluble. However, in the hydroxylation, the potency of the molecule may increase or decrease. Meaning to say, if you have a poisonous substance, the hydroxylation may increase. Certain molecules may increase its activity or potency. And some other molecules, when hydroxylated, its potency decreases. You get that? That is why we have to have another reaction in order to prevent this. And the next one is what? Phase 2. Phase 1 is hydroxylation. Phase 2.
Conjugation.
Conjugation. You conjugate it with something. No? To make it more water soluble so that it can be excreted through the kidneys, and also decreasing its activity or potency. Okay. Number 17. We know this already. Which reaction or final reaction prepares xenobiotics for excretion? And that is phase 2, and that is conjugation. No? Now, in the hydroxylation reaction of cytochrome P450, what is the required reductant?
NADPH.
NADPH. All right, NADPH, the reductant. NADPH. I want you to remember that because not only in cytochrome P450 but in other pathways later on, why this NADPH is very important. Now, NADPH is used as a reductant in synthetic reactions. Meaning if you want to build a bigger molecule out of smaller molecules, you require NADPH to put those two molecules together to become a bigger molecule. So that is synthetic process. All right, number 19. Cytochrome P450 are plentiful in which part of the cell? Lysosomes, Golgi complex, endoplasmic reticulum.
Endoplasmic reticulum.
Endoplasmic reticulum.
By the way,
All
Doc,
right.
by the way,
Yes.
in this part, Doc, in the exam, there was no endoplasmic reticulum among the choices.
Huh?
Yes, Doc. Maybe you can check that with Ma'am Shah.
But I have
bonus.
already,
The
well, if there is no endoplasmic reticulum, do you mean to say there are only two choices?
if we can remember, Doc, yung nasa A and B, yung lysosomes at Golgi complex, tapos yung C and D, Yung choice,
Walang D, C only.
basta yung next doc, yung choices niya yung from yung before na number 18, yun. Maybe you can check doc kay siya.
No, this is the copy, oh. Can you see this?
Yes, doc. Kasi kinaka-copy paste lang naman 'di ba nila? Maybe na
Oh, there. This is the one, oh. There. Well, anyway, I will ask siya, and if there is none, then that will be a bonus.
okay.
Naturally.
Yay!
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 Doon na ka na iwan.
Yes.
Alright, number 20. Glutathione. Glutathione is an antioxidant involved in the transport of certain amino acid into cells. All of A and B.
All of A and B.
You know it is an antioxidant, di ba?
Yes.
You know that. But what about the other one? Involved in the transport of certain amino acid into cells.
It's true.
Yes, Doc.
All of A
That
and B.
is correct. So both of them are correct. Number 21. Which of the following is not a xenobiotic, a foreigner, which is not a foreigner here? Penicillin, bile acids, pesticides, phenobarbital.
Bile acids.
Bile acids. All right, bile acids. Now, when a surgeon removes the gallbladder because of gallbladder stones, he does not have bile acids anymore. Di ba? And the surgeon prescribed bile acids or bile salts. Now, do you consider the bile salts now as a xenobiotic?
Yes.
No?
You say no.
Yes.
Okay, yeah, it's still no. That's correct. It's no. It is just like supplements like amino acids. If you have problems with your digestion of proteins and you cannot have so much of the amino acids. So, what is given to you is just plain amino acids so that they can be absorbed right away. So you do not consider that amino acids as a xenobiotic. All right. Or for example, if in type 1 diabetes, where your pancreas cannot make insulin. Now in type 2 diabetes, you have plenty of insulin. It is not correct to say that in type 2 diabetes, there is no insulin. There's plenty of insulin. It's only in type 1 diabetes where you do not have insulin. Now, if we give the patient type 1 diabetes insulin, you consider it as xenobiotic.
Yes.
It's a xenobiotic. Or it cannot also be a xenobiotic. Why? Because there are two kinds of insulin. Insulin coming from animals, either from pigs or horses, equine and porcine insulin, and human insulin. Well, that is not, of course, taken from human beings. What they did was to get the gene of human insulin
We
and
use
put it on a bacteria, DNA of the bacteria, and let the bacteria produce that human insulin. So it is not rejected because animal insulin, equine or porcine, you will notice that you will be increasing your dosage of insulin. Like for example, you start with 10 units of insulin every day. After a year or after 2 years, that 10 units is not effective anymore. So you increase it to 20 units. Now, why is that so? Because it is foreign. It is foreigner. No? And it is destroyed. Your immune system tries to destroy it. Maybe it will take them a long time to destroy. They do not have the capability yet for the first month, second month. But then when it comes to years already, they have now the capability to destroy it. And that's why you have to have a higher dosage of insulin. But with this human insulin, they call it human because the gene is taken from the gene of a human being and just transferred it to bacteria and let the bacteria produce that insulin and purify it. That is still human insulin because the codons there or the code for that particular protein insulin conforms to human insulin. There are differences in the amino acids in the animal insulin and human insulin. That's why that animal insulin are degraded. Oh. But it takes time to degrade it, meaning our immune system tries to gather its capability to be able to destroy it. So it takes time, 1 year, 2 years, before you increase your dosage. Okay? Number 22, cytochrome P450 is a multifunctional enzyme and can do the following reaction except reduction, hydroxylation, conjugation, dehalogenation.
Nation.
Halogenation.
Conjugation. It cannot do conjugation. Your cytochrome P450. Okay? Number 23. Now, you will notice I have only one question here which has an except. Because we are told to minimize except. But then we can still go around it. We can ask the question, which is not? So that is still exception, di ba? All right, the reductant, by the way, the APMC tells us to minimize the except in your questions. But the board exam, there's plenty of except. Sige na lang. The reductant used in hydroxylation reaction of cytochrome P450 is FeDH2, FnH2, NADH plus H, NADPH plus H.
C.
NADPH plus H.
Letter D.
Ready? Oh? NADPH plus H. 24. Cytochrome P450 is quite versatile biocatalyst. It uses a molecular oxygen and splits it into two atoms. And how are these two atoms of oxygen converted? Letter A, one atom of oxygen forms sulfur oxide and the other atoms form water. Letter B, one atom of oxygen forms hydroxyl and the other atom forms water. Letter C, one atom of oxygen binds with carbon to form carbon monoxide and the other forms water. Letter D, all of the above.
Letter
Boy,
B
boy, Doc.
forms hydroxyl and other forms water.
Letter boy, Doc.
Okay, that is forms a hydroxyl and water. One atom for formation of hydroxyl and one atom formation of water. 25. Aside from the xenobiotics, cytochrome P450 uses other substrates like retinoids, steroids, eicosanoids, all of them.
All of them.
Love them.
All of them. Your cytochrome P450 can act on retinoids. This is vitamin A, no? Steroids and eicosanoids. Number 26. Cytochrome P450 is so named because it was discovered A, to perform 450 reactions. Letter B, the 450th experimentation. Letter C, it can withstand a temperature of 450 degrees. Letter D, exhibits a distinct peak of 450 nanometer when reduced microsomes are exposed to carbon monoxide.
C.
D.
Letter C ba, Doc?
D.
D.
Letter D.
Pink. Pick up.
Letter D.
Meters.
Letter D. No? A distinct peak absorption in 450 nanometers. All right, another cytochrome, number 27. Cytochrome P3A is the most important cytochrome P450 because of its wide range of reactions, of its several isoforms, 600 are known, its abundance in liver and intestine, and its ability to use all known xenobiotics as substrates.
A.
Letter A.
It is important because it is so abundant in the liver and intestine. When you start the food that you eat, it's there already. And then plus the liver pa, na? Where it is the organ of detoxification. All right. Number 28. Which of the following is our characteristics of cytochrome P450? They are inducible. They have several isoforms. They are heme proteins. Letter D, all of A, B, C.
All of
All.
A.
Letter
All of them. Cytochrome P450 are inducible. That's why we usually have problems with drug interaction because of uh, being induced, can be easily induced. Letter B, they have several isoforms, which is correct also. And they are heme proteins, just like your myoglobin, just like your hemoglobin. Which of the glutathione not only play an important role as an intracellular reducing agent, na, this is a repeat question. Eh? they, they are involved in the, aside from that, they are involved in the transfer of certain amino acids into the cell. We have that question already, di ba? Kanina?
Yes, Doc.
Yes.
Nabalik. Pero sige lang. Ang problema lang nga na pareha raw ang tag-answer sa iyo pag yung usa. Number 30. The following are known conjugants used in phase 2 reactions in the liver for making synbiotics more water soluble. Except, so another except there, doa din except glucuronic acid, glutathione, phosphatase, sulfates.
Phosphatase.
Phosphatase.
Phosphatase is an enzyme. No? You have your conjugants, glucuronic acid, glutathione, and sulfates. 31. Membrane proteins that span the whole width of the membrane are also known as
Integral.
Integral po. Balik.
balik. Which of the following statement is true? All diseases are caused by bacteria and viruses. All diseases are due to ability of the immune system to contain the microorganisms. All diseases are caused by changes of the environment. Most diseases have a biochemical basis.
D.
B.
D.
D. That is a subheading, di ba? Heading of a topic, of a paragraph in your book. Most diseases have biochemical basis. 33. COVID-19 is caused by which of the following? Physical agents, chemical agents, biological agents, genetic disorder.
Biological agent.
Letter C.
Biological.
Which omics is the RT-PCR test for COVID-19 belong to?
Letter B.
Transcriptomics.
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 Transcriptomics. 35. What property of water most contribute to its ability to dissolve compounds? What property of water most contribute to ability to dissolve compounds? A. Absence of interacting forces. B. Strong covalent bond between water and other molecules. Letter C. Hydrophobic bonds formed between water and long-chain organic acids. Letter D. Hydrogen bond between water and long-chain biochemical molecules.
Dog ba?
D.
Letter D.
Letter D. Because of its hydrogen bond. Ah, if humans are made of or are made up of 60% water, what is the total volume of water has a 50 kilogram woman? 10 liters, 20 liters, 30 liters, 40 liters.
30? 30
30.
liters po ba?
How did you get it?
50 times 0.60.
60% of 50. What is 60% of 50?
30.
30.
30 kilograms. Di ba? Kay 50 kilogram woman man eh.
25.
Yeah, 60% water. 60 times 50 is 60% of 50 is 30 kilograms. So what's your answer?
30.
30 po, Doc.
30
30
liters.
po, Doc.
1 kilogram is 1,000 cc and 1 cc equals to 1 gram. Di ba? 1 cc of water is 1 gram. Yeah, 30 kilograms. Ilan na? 30,000. 1 kilogram is 1,000, di ba?
Ako.
Oh. 1,000 cc.
Mag-message.
So that's 30 liters.
Nasend na kaya niya ha email?
Water molecules self-associate via disulfide bonds, hydrogen bonds, peptide bonds, glycosidic bonds.
Hydrogen bond.
Hydrogen bond.
Hydrogen bonds.
Hydrogen bond po.
Which of the following has the highest bond energy? Oxygen bonded to oxygen, carbon bonded to carbon, carbon bonded to hydrogen, carbon double bonded to oxygen.
Carbon double
Double
bonded to
bonded to
oxygen.
Oxygen.
Double
Letter D.
band. Letter D.
Double bond.
Number 39, amphipathic compounds possesses regions with polar and nonpolar groups, regions with positive and negative groups, hydrophobic groups and negatively charged groups, acidic groups and basic groups. Which
Letter
one?
A po.
A.
A.
A. Regions with
Polar and
polar
polar.
group po.
All right. Electrostatic interactions between oppositely charged groups within or between biomolecules is called van der Waals, peptide bonds, salt bridges,
Salt
SSBA,
bridge.
salt
Salt
bridges
Salt
bridges.
bridges.
interactions or ionic interactions. Huh? Okay, so that's what is the percentage of passing there?
A.
See?
30, Doc.
20.
Were you able to answer Dr. Ryan's questions?
Slight lang do.
Light lang, Doc.
Come lang,
Right,
Doc.
Doc.
Right daw.
Sakto lang,
Ilan?
Doc.
Sakto lang.
Okay, sakto ra. O niya, sakto ra sa din sa ito pa, patar na. Okay. Now, I don't know if I will be lecturing tomorrow. I don't know whether Dr. Romel is ready. If she's not ready, then I'll be the one lecturing tomorrow. Yes, so is supposed to lecture on physiologic carbohydrates. Carbohydrates of physiologic importance and lipids of physiologic importance. Then we go into what you call this introduction to metabolism. Introduction to metabolism. Now, I don't know if, have you had your red blood cells and white blood cells lecture? Not yet.
Done na po, Doc.
Histology
Doc.
po.
In
Done na
physio?
po.
In physio?
Histo.
Histology
Oh.
po.
And also here in biochem.
Also histology. And also Dr.
Yes.
Barito,
Yes,
Yes
Doc.
po.
It's already
Yes po, Doc.
red blood cell and plasma. No? Proteins.
done. Yes, Doc.
Okay.
Yes po, Doc.
So it all depends now on Dr. Romel Kapungkol. If he is ready by tomorrow, then he will be the one to lecture. So if he's not, then I will just give you a bird's eye view of metabolism. Because you have already finished your enzymes, so we will go into metabolism. Is that okay? Huh?
Yes po.
Okay
Yes
na.
po, Doc.
Yes,
Yes,
Doc.
Doc.
Yes po, Doc.
All right.
Yes, Doc.
Oh, tapos na ta.
Yay.
Thank you, Doc.
Salamat po,
Thank you,
Doc.
Doc.
Welcome.
Thank you, Doc.
God bless.
Thank you, Doc.
Thank you, Doc.
Doc, thank you.
Salamat, Doc.
And, hmm,
Thank you, Doc.
welcome.
Bye, Doc.
Thank you, Doc. Bye, Doc.
Thank you, Doc.
Ingat ka
Doc,
palagi,
thank you,
Doc.
Doc. Thank you.
Thank you, Doc.